Good morning. It is my great pleasure today to introduce Helmut Kulfen, who's going to talk to us about his fabulous prenucleation clusters. So Helmut started his career as a physical chemist in Germany at the University of Duisburg, where he got his PhD in 1993. Then he worked for a long time at Max Planck Institute, where he went up the food chain, ending being group leader. And then most recently, he became a full professor, just moved this past fall at the University of Constance in the south of Germany. So he's just in the middle of moving his gigantic group, his gigantic lab, and making it all work there. So he did his PhD thesis on analytical ultracentrifugation of gels, and then he went off and actually discovered mesocrystals and how nanoparticles attach to one another a la Banfield and constitute these large arrays of co-oriented nanocrystals. And he found 5 different pathways for that. He got more than 6,000 citations. His H-index is astronomical. But then he went back to the origin, back to analytical ultracentrifugation. And the most recent thing that made him famous are these prenucleation clusters that he's going to tell us about today that he published in Science just a couple years ago. Thank you.
Thank you very much, Pupa, for this nice introduction and for the opportunity to be here at this place. So I'm enjoying Berkeley already for the last days. So it's great to be here. Pupa already announced it. The topic of my talk today is centered around calcium carbonate, and I'm going to focus on prenucleation clusters and the very early stages of nucleation and how you can control it by additives. So basically, the pathway, which is an alternative pathway to the classical nucleation, is already outlined here, that you start from supersaturated solution, then go via clusters, form amorphous calcium carbonate, which nucleates and then finally forms the crystal. And I'm going to show you more details about that later. So first, let me start why calcium carbonate is important. And I guess many of you work in the topics of carbonates, I figured out last days. So it's certainly important because it's got big geological deposits and binds enormous amounts of CO2. And it's also important because it's the most abundant biomineral. So you find lots and lots of biominerals, shells, snail shells, corals, algae, and large deposits of these biominerals too. And it's very intriguing to learn about biomineralization, as I will show you on some of the next slides. And it's got a great industrial importance. I was just shocked. I was at the PAsp meeting a few weeks ago, and I learned there that 40% of the CO2 production worldwide is just done by burning calcium carbonate to produce calcium oxide, basically, and that consumes 30% of the world's energy. So it's a real big issue, and it would be great to learn more about alternative processes not to produce so much CO2. It's also used as a pigment, filler in paint, paper, plastics, and so on, and most importantly, the major component of cement. Also, we all know scale in water pipes, and also calcium carbonate is important here too. So scale prevention is industrially a very important issue. And it's, last but not least, a very good model system. It's got ionic bonds. It's not too complicated. It's got several polymorphs. And the anhydrous ones are calcite, which is stable, aragonite, metastable under ambient conditions, and vaterite. So one very important occurrence of calcium carbonate is certainly in biominerals. And it's very fascinating to study them. So I devote 2 or 3 of the next slides to it. And one important system is certainly Nacre. So you see this inside of mussel shells. You got these iridescent colors. And it's a very hard material, so I don't have a mussel shell with me, but believe me, if I would smash it here on the edge, it won't break, but probably the edge might somehow be damaged. And the reason why it's such a nice material is because it's layered. So you have calcium carbonate tablets in the form of aragonite, and they are fully surrounded by organic layers. So you have, if you dissolve out the calcium carbonate, you have these organic layers made out of chitins covered by proteins, and you have these bridges in between the layers. So every hard tablet is fully covered by the organic layers. And if you look at developing nacre, you see that first the cells secrete these layers, and we now know that they are filled with a silk hydrogel, and then the calcium carbonate tablets start to grow there, and then later on meet and form this material. So this is a typical stack of coin architecture, And here you see the tablets. When they are grown together, they are interspaced by an organic layer. And that adds to the nice materials properties. So if you look schematically, Nacre is something like a brick and mortar architecture. So we have these soft organic layers which dissipate the energy. And if you compare that to man-made buildings, concrete or gypsum, this is a messy architecture. And that's why concrete and all these building materials are not so strong and tough as Nacre, because they are not organized. So if you look at Nacre when exerting a force, then you will certainly break the first inorganic tablets, which adds strength to the material. And then you come to the first organic layer, which you have to tear apart, that dissipates energy, and so forth. So you also prevent cleavage planes, which you have in a single crystal. And Nacre is even more complex. So the inorganic crystals are glued to the organic layers by proteins, which have sacrificial bonds, which also dissipate energy further. So if you pull at them, the bonds may loosen and they may stretch. So that's another mechanism to dissipate the energy. But the big question is now, why are these biominerals so different from a normal crystal, and how do they form? So I have calcium carbonate here as a geological deposit in the form of calcite, the typical rhombohedral crystal. We all see it's got a simple shape, it's got faces, and it's got angle between the faces. So that's what we know from a crystal in a textbook. It's got no hierarchy, and usually it's formed under harsh conditions, and it's purely inorganic. On the other hand, if you look at calcium carbonate as a biomineral, and this is calcite 2 in the form of a sea urchin spicule, you see, if you zoom in, and this is the scale of microns, get a scale bar here, that you don't have crystal faces anymore, and the material's got a spongy appearance. It's a hierarchical material, and usually biominerals are evolutionary optimized. That's why they have such nice properties, and they are formed under ambient conditions, and they are usually organic-inorganic hybrid materials. In that case, it's 0.1 weight% of organics, which is in there, so it's mainly inorganic, but this little amount of organics makes up nice properties of this biomineral. And if you now zoom in further, you can see at this cleavage plane that you have nanoparticles, and then the big question is, how do you start to grow this material? And this should start with the very basic question, how does a single crystal nucleate? So if you ask yourself that question, then you can go to the textbooks and come across the classical nucleation theory, and this is basically shown here in the sketch, where you make up a nucleus by investing surface energy. So if you precipitate something out of solution, you create an interface between solution and your crystal in that case, and that creates surface energy. So if you assume you got a spherical nucleus, then this is the term for the surface energy, and that scales with the radius of the nucleus squared. On the other hand, you win bulk energy, and that's the blue curve here, and this scales with the radius to the power of 3. So that's the energy win, and if the nucleus is very small, then the surface term wins over the bulk energy term, and that means you have to invest energy, and it's not a stable situation, so the nucleus will fall apart again. But once it comes across a situation where you just balance the surface energy with the bulk energy, and that's the so-called critical crystal nucleus, then you start to win energy if the crystal grows, and that means now the crystal can grow, starting from the critical crystal nucleus, and then become even macroscopic. So you get particle growth beyond a critical size. And this growth is known to occur in case of a crystal via steps. You can beautifully image that by AFM, and Jim Dioreo is very famous for producing very nice videos how a crystal grows here. And you see that you can get steps, you can get kinks, you can get surface vacancies, and you can get growth units attached to a face which will then diffuse and then eventually attach to a step or to a kink. So finally the crystal grows layer by layer until you end up with a macroscopic crystal. But if you think about biominerals, you have some biological constraints. And one big constraint is that it's usually taking place in a living system. That means cells are involved. So if you want to crystallize something in cells or outside cells, you'll have to think about the ionic strength. So if you increase the ion concentration to precipitate the crystal, you will also create an osmotic pressure. And that may be even so big that the cell may burst. So it may not always be very advantageous to crystallize a mineral from ionic solution. The other thing is, if you've got a sparingly soluble salt and you want to crystallize that as a biomineral, you have to transport large solution amounts through the cells to sequester enough ions to get over the supersaturation limit to finally precipitate the biomineral. So that's also not advantageous. So that results in the fact that ion accumulation can take a very long time until you precipitate something in a natural environment. On the other hand, if you have precursor phases, which are built permanently and then are transported to the building site, and this is known for biomineralization, for example, in forms of vesicles, which were detected in sea urchin spicules, then you prevent all these high ionic strength as a building site inside the cells or outside the cells, and you're just working with a precursor phase transported to the building site and assemble the mineral. So that's obviously advantageous. So it makes sense to think about particle-mediated mineralization or amorphous precursor phases, which are detected by many groups in biominerals in the last years. So it makes sense biologically. And if you look at the process of crystallization, then you have a whole zoo of species which can occur. And I've taken that graphic from a nice review of Jens Rieger and Dieter Horn in Angewandte 10 years ago. And they looked at the species which you have in a crystallization event. So you start from a homogeneous solution. So these are usually ions if I consider calcium carbonate. And you can run through precursor phases. And you see here, existence question mark. So do I have precursor phases, yes or no, until I arrive at a critical crystal nuclei, which can be amorphous or crystalline. The structure also question mark, because it's very hard to investigate that analytically. And then you come to the intermediate stages where you can have growth of amorphous structures, crystalline structures, hybrid structures, or aggregation or stabilization of nanoparticles until you end up with a zoo of final species which you get in a mineralization event. And we were asking ourselves the question, what happens at the earliest stages, even before we form a critical crystal nucleus? What happens if I look at the ions on the pathway to the final crystal? So what is this? And the experiment we performed is a very simple one. So if you take a beaker and you fill it with a carbonate buffer and you put it into a system which is very controlled by a titration machine, which keeps the pH constant to the third digit, and then you start adding calcium chloride solution into the carbonate buffer by a syringe pump at a constant rate, and we measure the calcium concentration via the potential, and as soon as we form calcium carbonate in here, the pH will drop and sodium hydroxide will be added to keep the pH constant. So we get a reading of the carbonate production via the consumption of sodium hydroxide, and we directly measure the calcium concentration in solution. And if you do that, you get curves which are shown here. So you measure, for example, the calcium concentration. You can also plot the carbonate concentration consumed, if you like, to versus time. And you get a curve which starts increasing linearly with the calcium concentration until you get to the nucleation point here, to the maximum, where nucleation occurs. And that leads to a dramatic drop in free calcium concentration up to a level which is constant, and that is the solubility product of calcium carbonate in that case. So you have three phases here. We have the pre-nucleation phase, before nucleation occurs, and then after nucleation we have the post-nucleation phase. And it's important also to consider that the level after nucleation defines the undersaturated region at concentrations lower than the solubility product, and the saturated region where I have a thermodynamic driving force towards crystallization. And it's important to say here in the undersaturated region, I don't have any thermodynamic driving force for crystallization. So crystallization is not expected to occur in the undersaturated region. And it's also important to consider that up to the point of nucleation, I'm really working in two solution. So it's a one-phase system. And after nucleation, I have a two-phase system. So I have, for example, amorphous calcium carbonate in equilibrium with the ions in solution. So here, a one-phase system, and after that, a two-phase system. And if I do titration experiments like that at different pHs, I will influence the carbonate-bicarbonate equilibrium, which is clear. So the higher the pH, the more carbonate I will have in my equilibrium. And you get curves like that. So you see they are rather reproducible. The nucleation point varies, and also the supersaturation at nucleation, because that's a stochastic event, because I have to jump over an activation energy barrier to nucleate, and this is statistical. So the nucleation point is not always at the same time. But what you see is that depending on the pH, I get curves like that. And the higher the pH, the lower is the calcium concentration in solution, what I would expect because I have more carbonate and more calcium carbonate formation. And the important point now is, if I compare the calcium which I added, and that's the white curve here, to the calcium which I detect in solution, I always detect the difference. And more importantly, if you define the solubility product here, you see that the difference is there straight from the beginning in the undersaturated solution. That means even when I don't have any driving force for crystallization or nucleation, I already have a difference between detected and added calcium. And that difference becomes even bigger the higher the pH. And it can be up to 75% at pH 10. That's the magenta curve here. So that means most of the added ions are not single ions in solution anymore. And the question now is, where are these ions? Are these all ion pairs? Is there something bigger, or what happens? So we took the solution out of the vessel and put it into analytical ultracentrifuge at different times. So these times represent increasing calcium concentration. So if you start only with a carbonate buffer, this is the white curve, and shown here is the sedimentation coefficient distribution. The bigger the sedimentation coefficient is, means the bigger is the species and the more dense is the species. So on the right side here we have the bigger species, here we have the small species. And for the carbonate buffer, we just detect carbonate ions. That's the white curve here. After 10 minutes, we have added a tiny amount of calcium. You still see only ions. 20 minutes, the green curve too. But then after 40 minutes, we see ions as well as a larger species. And as you will see later, these are clusters in solution. And you can repeat that experiment. That's the dashed curves. And you get the same finding again. And you see a little experimental variance because it's at the limit of the centrifugation. Because if you really think about the experiment, we are separating ions from something which is not much bigger than ions. So that's even at the limit of an analytical ultracentrifuge. We can also do electron microscopy of that. And I show you this. This was done in the lab of Nico Sammerdijk in Eindhoven. And he looked at this solution, which I've just shown you, to contain clusters. And if you look at calcium and carbonate solution already after 1 minute, and that's even in the undersaturated state, you see tiny spots. I zoom in later, but I circled them for you. Very tiny species, 1 to 1.5 nanometer big, and they start to aggregate with time. And after 24 minutes, you see they are obviously larger aggregates composed of little particles. So you see it in the zoomed image. Here you get a pretty well-defined species, defined in size. And they get more and more, already start to aggregate. And then finally you have aggregates. And if you then further add calcium to carbonate solution, the concentration of the species gets even higher, and you precipitate macroscopic or microscopic species of a few microns in size. So that's light microscopy image. And if you look at it under cross polarizers, you see that all the particles are black. That means they are not birefringent, and that means it's amorphous calcium carbonate. So the story here is we have little clusters, which we can detect even in undersaturated solution. They aggregate and then form amorphous calcium carbonate, which later on will crystallize. So the sketch you can make out of that is that we obviously have an alternative pathway to the classical crystallization pathway. So we start from a supersaturated solution, and in the classical pathway, you would form ion aggregates, and as long as they don't reach the critical size, they will fall apart because the surface energy is higher than the bulk energy win. And only if they exceed the critical size, they will form a post-critical nucleus, which then further grows to the final crystal. Whereas the pathway I've just shown you is such that I have ions. They form stable prenucleation clusters, which are pretty defined in sizes. We don't know so much about their mass yet, but they aggregate if you increase their concentration to amorphous calcium carbonate, which nucleates and finally forms calcite. So it's an alternative pathway. And if you plot it in an energy diagram, you can see it like this, that we start with ions, And finally, we have solid calcium carbonate, in our case, amorphous calcium carbonate. So the classical way would be that I form metastable clusters. They fall either apart or they can grow. And the alternative pathway, which we have now, is that we got a minimum before we form amorphous calcium carbonate, which is stable towards the ions in solution, and via activation energy, it forms amorphous calcium carbonate by passing this activation energy barrier. So that's the pathway. And it's very interesting that these clusters already seem to have different structures. So if you look at different pHs, you can describe these clusters by a binding equilibrium here, and this is a formula for it. And from that, you can get an equilibrium constant for ion pairing and cluster formation. And if you do that, you can plot the free enthalpy of ion pair formation versus pH. And you see at a lower pH, I have the most stable clusters, and these guys finally end up in calcite, whereas at a pH of 9.75, I get a situation which is less stable, and they form vaterite. And here are the titration curves plotted. And if you look at the solubility products, which you have here, compared to the crystalline polymorphs, you see that they clearly differ. So at the lower pH, I have a lower solubility product. This is amorphous calcium carbonate 1. And at a higher pH, I got more soluble species. And usually, if I got different solubilities of particles or species, that hints at different structures, although these clusters are certainly amorphous or X-ray amorphous. And Dennis Gebauer did, as a postdoc in Stockholm, some experiments to precipitate these prenucleation clusters by quenching them with ethanol, dehydrating them very fastly. And then he was able to look at them with EXAFs and solid-state NMR. And you see the results here. So with solid-state NMR, if you have the clusters which are formed at high pH, these are the white curves, they show a curve which is similar to vaterite here, that's the dashed curve, whereas those ones at a lower pH, the yellow curve, are consistent with crystalline calcite. So that implies that the structure is already encoded in the prenucleation clusters, and there's similar information from EXAFs. I don't know much about this technique, but people told me that already the structure of vaterite or calcite is also encoded in the EXAFs traces. So the message here is that not only ACC, the amorphous calcium carbonate, already contains a local order which corresponds to the final crystalline polymorph, but apparently already it's encoded in the prenucleation clusters, which are just a cluster of a few ions. And then we tried hardly to characterize the clusters themselves. So the results Dennis got in Sweden are nice, but they were done on amorphous calcium carbonate, which were precipitated from the clusters. And you never know what happens if you add ethanol, because you dehydrate them. I will later show you that you get rid of 19 water molecules per calcium carbonate molecule. So that may change their structure. So it would be much better to look at the clusters themselves. But unfortunately, from ultracentrifugation, I forgot to mention that, we got the information the cluster concentration is very low. It's only 0.1 to 1% of the total ion concentration is present in clusters. So most of it's either ions or ion pairs. But it would be nice to stabilize them and enrich them. And Matthias Kellermeier from the University of Regensburg did some experiments by precipitating these clusters in presence of silica. And finally, what he ended up with was clusters which are coated by a silica shell. And you see high-resolution TM here, and you see the clusters all over the space. So if you zoom in here, the rectangular thing, you see the clusters are there. They are pretty defined in size. They are about a nanometer big. Here they are a bit more polydispersed than what Nico Samardijk got, but this is understandable because they are covered with a silica shell. And if you wait, then the clusters aggregate with time. So this is a light scattering experiment. You see the increasing intensity with time. This means clusters aggregate. You can see that in cryo-TEM. And you also see that the solution becomes or starts to get opaque. So these are aggregates of about 60 to 100 nanometer in size. And you can see them here, but they are composed of individual clusters. Now, if you increase the silica concentration, then you are able to stabilize the clusters and to prevent the aggregation. And you see some dynamic light scattering experiment here. So you have the prenucleation clusters. Then you have a first sort of aggregates, which is 9 nanometer big. And then you got about 60 nanometer big larger aggregates. And And if you take that solution and centrifuge it, you can take away the large aggregates, and then you are only left with the prenucleation clusters and their first aggregates, which are about 9 nanometers big. And we are currently working on trying to characterize what the structure is. Although here, again, we have an obstacle. Who tells us if not the silica is changing the properties of the clusters again? So, in fact, the clusters are an equilibrium species, and we are now just freezing them in by covering them with a silica shell. So, let's see what the structure is. Nevertheless, the clusters seem to be much more common than we initially assumed. So, first we looked at different biominerals. So, for example, we did the same experiment with calcium oxalate, and that's the yellow curve here. And again, you get the same story like calcium carbonate. You get formation of clusters, which you can see in the ultracentrifuge. You can do the experiment with calcium phosphate, that's the red curve, and you see cluster formation again. And Nico Summerdijk published a few papers quite recently, beautifully showing the clusters in electron microscopy for calcium phosphate. So it seems that at least for biominerals, the clusters are relevant. But we did a few further experiments. So first we started to look at sodium chloride in the analytical ultracentrifuge because we wanted to have a system which does not form clusters to compare it. But then we were quite astonished, and we saw, you see here the columns, that sodium chloride also forms clusters and cluster aggregates. And you have here a whole bunch of different salts. You have soluble salts, you have very sparingly soluble salts, and I don't want to go through them, but you see that they all show ions in the analytical ultracentrifuge, and they show clusters. The only species we didn't see clusters so far was guadinium hydrochloride, a strongly structure or water structure breaking agent. Oh, that's the only one which didn't form the clusters. And you can also see in a series of, for example, iodides, if I make the cation bigger, go from lithium to cesium, then also the clusters got bigger. But this is no rule. So if I take the carbonate, starting from magnesium carbonate, going via calcium to strontium carbonate, first the clusters get bigger, but then smaller again. So it's obviously no rule that the cluster size or sedimentation coefficient scales with the size of the ions. But these results imply that cluster formation is a much more general phenomenon. At the moment, we are working on a fast screening technique to look at ideally hundreds of different minerals to see whether they form clusters or not. But so far, for the few systems we look at, we always found them. Then we did some calorimetry to work out the thermodynamics and to get some information about driving forces. So this is an ITC experiment by titrating calcium ions into a carbonate buffer and then measuring the heat. And you see that always it's endothermic, so heat is consumed. We tried to fit these curves and were not successful at all. We used two models. One model was ion pairing. The other one was a multiple binding model by going from ions to clusters. Both don't fit these ITC curves. We're still working on it. But what we can certainly say is that cluster formation and ion pair formation in the sum of the system is endothermic. And if you look at the well-known equation of the free enthalpy or free energy, which is, must be negative because ion pairs and clusters form, and we know that the enthalpy is positive, then we must have an entropy release. We must have an entropic driving force. So the question is, what is this driving force? What drives the ion pair and cluster formation? And for this, we did an experiment in the analytical ultracentrifuge to work out the density of the clusters to get information about the water content, because we suspected that it's water release which drives cluster formation. And the experiment is such that we repeat the experiment detecting clusters in solution in water and do the same experiment in D2O. And the assumption is that the cluster structure doesn't change, which is the underlying assumption here. And then you can work out the density as well as the size. And if you compare the density of the clusters with the density of amorphous calcium carbonate, which was determined by small angle X-ray scattering by Balov a few years ago, you see that it's much lower. So that means clusters contain a lot of water. If I know the density of the clusters, I can work out the size, which is about 3 nanometers. That's bigger than what you get out from electron microscopy. Molar mass is about 4,000 grams per mole. That means you have about 40 calcium carbonate ion pairs in the cluster. On the other hand, information from Nico Sammerdijk, cryo-TEM on the clusters is that they are much smaller. So you can imagine two structures. Either you have ACC core with associated hydration water, or more likely you have a homogeneous structure of bound water and calcium carbonate. So we can't distinguish the structure with the present data sets yet. But what is important is that if I have the density of the clusters, I can work out via this simple equation how much water I have in the cluster, and the number is very high. It means 75% of these clusters are water, and that means it's more than 20 water molecules bound per calcium carbonate. So essentially what we are looking at is water. And the estimations which underlie this calculation is, because I work in D2O, the real density must be even lower. That means the real amount of water must be even higher bound to the calcium carbonate. We assume that the density of amorphous calcium carbonate in the clusters is the same as in bulk ACC, which we don't know. So it should have the same structure. And we say that the hydration water density should be the same as the bulk water density, which may also not be justified. And we assume that the cluster structure is equal in water and D2O. So there's a few assumptions going into that number, but nevertheless, it's at least an estimation of the amount of water. And now you can go to the literature and look at published values of hydration water, of ions. And when you do that, you can find various informations. For example, calcium has about 6 to 7 water molecules per calcium associated to it in the first hydration layer. Carbonate has 9. And then in the second layer, the numbers are varying. And for carbonate, I couldn't find anything. But if you add them up, you can say that we have 16 water in the first layer and about 22 water molecules in the second hydration layer, if I assume that carbonate is hydrated as calcium. And that means I got in total 38 molecules of water surrounding the ions. And if you remember the number of 20, which we have found for the clusters, means I release about 18 water molecules by coming from the ions via ion pairs to the clusters. So there's a significant release of water, which may be the driving force for their formation. So here's a summary about the clusters. So they have a size of about 3 nanometer in case of calcium carbonate, pretty defined in size. Molar mass is about 4,000 gram per mole. Concentration is pretty low, 0.1 to 1% of the total ions. So most of the ions are present in ion pairs. Their formation is endothermic, so you have to invest energy to form the clusters. So that means because they are forming, they must be driven by entropy. And the reason for that can be the release of 18 molecules of water per molecule of calcium carbonate, which forms an ion pair and a cluster. And if you just estimate the energy, it's quite significant. It's 67 kilojoule per mole at the maximum. So a significant entropic driving force for cluster and ion pair formation. Now, if you go from the clusters to a more complex system and now start adding additives, which is the relevant case in natural environment usually. So you have all sorts of other ions which can add as additive to change the formation of calcium carbonate. Then it's very interesting to look how these additives act at the very early stages until nucleation and after nucleation. And you can get typical curves. So if you remember the titration curve, the white one is the default curve, where you increase calcium concentration, you have nucleation, and then form amorphous calcium carbonate. So the first thing which can happen is that you have an additive which can absorb calcium ions. And you detect that via a curve like that, shown in the red curve, that every calcium ion which is added is bound to the additive. So you don't have free calcium in solution until the additive is saturated, and only then the curves start to rise. And then you have a second point, sorry about that, where you have nucleation, which is delayed compared to the reference experiment. So in that case, you can have an inhibition. You can also change the equilibrium of clusters. So if you remember that the amount of ion pairs and clusters is defined by the difference between added calcium and detected calcium, a lower slope of this curve would mean that I have more ion pairs formed or more clusters formed. So you can change the cluster equilibrium by additives. You can also delay nucleation and increase the supersaturation for nucleation. And you can finally form a more soluble species than in the reference experiment. So in the reference experiment, we had amorphous calcium carbonate, and And I will show you later on there are some additives which can even form something more soluble. These are the liquid precursors which were found by Laurie Gower. So here's a real measurement curve. The little white guy here is the reference experiment. That's normal calcium carbonate without any additive. And if you add polyacrylic acid, which is a well-known scale inhibitor, so polycarboxylate, which will bind calcium, then you will see it's already binding calcium straight at the beginning. So if I add calcium, it's bound. And I can quantify that by extrapolating the curve. I know exactly how much calcium is bound by polyacrylic acid. Then the curve rises. And you see that nucleation is significantly inhibited in time compared to the reference experiment, and also in supersaturation. So the calcium concentration at which nucleation occurs is much, much higher in presence of polyacrylic acid. Then there's another feature. You see that the slope of the curve changes at a certain time. So that means the cluster equilibrium is changing. And I get also a second and a third nucleation event. That means as soon as I nucleate, in that case amorphous calcium carbonate, it releases some of the polyacrylic acid, which then nucleates again and nucleates again. So it's a complex behavior of an additive, which you already see from the curve of that very simple scale inhibitor. So we can hope to extract some characteristic features of additives. And I will show you later on that each additives behave differently. And then perhaps we will be able in the future to use these features and predict what the additives are doing. So I walk you through the action of some of the additives. So if I look at the precipitates of calcium carbonate in presence of polyacrylic acid, in the reference experiment at a high pH, I get predominantly vaterite and some calcite crystals. If I add polyacrylic acid, I form pure phase calcite. So obviously it's also got an influence on the polymorph formed, and that was interesting to us, why these additives can influence the polymorphs. If you plot the solubility product versus the time, you get this curve like the white curve. So increase the solubility product, you are oversaturated, supersaturated, you have the nucleation. And then you see the first nucleated species has a solubility product of the pre-vaterite amorphous calcium carbonate. Then you get a second nucleation event. Again, it's a solubility product of vateritic cluster, but then it falls down to the more favored calcitic clusters. So obviously, polyacrylic acid stabilizes the vateritic clusters, and the not stabilized calcitic clusters later on form the amorphous calcium carbonate, which then forms calcite in the end. And the same we found when looking at peptides. So we did some work on 3 peptides which were published by Angie Belcher some years ago, and these peptides were found by phage display as adsorbing on aragonite surfaces. And we were interested in the question whether these peptides would nucleate aragonite. So they had a lot of basic chain amino acids in there, which are displayed here in green. And we know that these peptides have a binding affinity most to aragonite, then to calcite, and then to vaterite. So now if you look at the titration curves at a pH of 9, where you have usually calcitic clusters, you see that the 3 peptides show some differences. So the green peptide 2 has the highest nucleation inhibition, whereas at pH 9.75, where you have vateritic clusters, the peptides don't differ that much. So that means that the nucleation of calcitic cluster is much better inhibited by peptide, the green, peptide 2, and this magenta one, peptide 3, than for the calcitic clusters. And that means that we have pure phase vaterite formation in presence of peptide 2 and 3, because the calcitic clusters are inhibited more than the vateritic clusters, which later on nucleate vaterite. So that can be a handle how to control the polymorph by inhibiting those clusters and stabilizing them, and then those non-stabilized clusters will form the polymorph, which you find in the end. That's at least true for polyacrylic acid and these peptides. So now let's have a look at different additives, and a very simple additive known to be a scale inhibitor too is citric acid. And you see here with increasing citric acid concentration, that's the reference curve, see that nucleation is inhibited more and more. You also see that the slope is changing. So I have a binding of calcium ions in the early stages due to complex formation. I get some inhibition of nucleation, and I get a smaller slope of the linear part. That means the cluster equilibrium or ion pair formation equilibrium is changing. So with increasing citric acid formation, the slope gets lower, so it's decreasing. I'm sorry for these busy slides. That's another additive. That's sodium 3-polyphosphate. That's a strongly calcium-binding agent. And here again, you get a concentration series. And I just would like to ask you to focus on the red curve here. So that's just done in repetition, that you see it's fairly reproducible. And again, you see that you increase the concentration. The slope of the curve is changing. That means the cluster equilibrium is continuously changing. You get inhibition of nucleation with respect to the reference curve here. And then you form a first phase, which is much more soluble than amorphous calcium carbonate formed in a reference case. Then you get a second nucleation event, and then the solubility drops to that of amorphous calcium carbonate. But you form something in between, which is much more soluble. And this is a liquid precursor, as I will show you later on. So the same happens if I look at polyaspartic acid, which is known to form these precursors. Here in white, we have the reference curve again. So if I increase the concentration of polyaspartic acid, I get a slight nucleation inhibition. And importantly, I get a species which is nucleated, which is much more soluble than amorphous calcium carbonate. And this is really, when you take a look at the sample, then you observe droplets. This is taken from a paper of Laurie Gower. These are the so-called polymer-induced liquid precursor phases. This means some stuff which you get at a low polymer concentration in the microgram per milliliter range, where you have precursors which are liquid, and then they eventually crystallize, and you can form very, very complex shapes. So there's a lot of work done by Laurie Gower on this, and here we now can trace that for the first time. So after nucleation, we see something which is much more soluble than amorphous calcium carbonate. It also changes the composition. So you see that too. And that's also something Laurie Gower found. So the liquid precursors are never the same time-wise. So they change their composition until they nucleate. In the end, you get amorphous calcium carbonate, which then crystallizes. So you can directly see it quantitatively in this titration setup. Another additive is one which forms mesocrystals. So that's polystyrene alt-malic acid. And this one behaves differently. So you see you got ion binding again. Also, you get nucleation inhibition. You get nucleation of a precursor phase, which is more soluble. And if you look at the crystallization products, later on, this guy or this additive forms mesocrystals. I don't want to go into the formation mechanism, but you see here, this is calcite. It's self-similar. This is a triangular phase, which is the O1 phase stabilized by this additive. And you see each of these triangles composed of subtriangles and subtriangles and so forth. So this is an additive which forms superstructures. And the pathway it goes is that you start to go from nucleation clusters via primary nanoparticles, which get stabilized by the polymer and finally form these polycrystalline superstructures with perfectly crystalline aligned nanoparticles. So I just want to flip you through the additives I've just shown you. We have more curves now to show you that additives have a very characteristic profile. And we hope to extract the features and get out the fingerprint regions basically to make predictions in the future. So here we have calcite. You see the course of polyaspartic acid is very different. This is magnesium ions. They also act as additive. Sodium 3-polyphosphate, polystyrene malic acid. And this is a commercial scale inhibitor. This is polyacrylic copolymer with styrene sulfonate. And again, a very different behavior. So if you overlay them, you see you got a whole zoo of different curves which tell you something about the action of the additive before nucleation, upon nucleation, and after nucleation. And I think we haven't even extracted all the information which is in these curves. So they're very characteristic for the different additives. So this is a topic of current work. So the last few slides, I would like to show you a last experiment which is relevant to biomineralization. And this is a question that many biominerals, calcium carbonate formed in seawater, is in the form of magnesium calcite, but the magnesium content is rather low. So it's only a few weight percent, at the most maybe some 20 or 30 weight percent, but not more. And if you look at the composition of seawater, you have an excess of magnesium. So it's a 5-fold excess of magnesium over calcium. And the question is, why does not more magnesium precipitate in the form of biominerals. So what happens upon formation of these calcium carbonate deposits in biominerals in seawater? And where's the magnesium going? And one intriguing thought we had, if there's prenucleation clusters, may it be that the prenucleation clusters expel the magnesium and just like more calcium carbonate formation rather than magnesium carbonate? So we set up an extension of our titration experiment and added a second electrode, which is magnesium sensitive. So we are measuring magnesium ions in solution, calcium ions in solution. We added calcium chloride and magnesium chloride continuously, and we kept the pH constant. So it's the same experiment now, just extended for magnesium carbonate detection. And the curves you get are similar to what you know from calcium carbonate. So now we detect the free magnesium. This is the magnesium added, the white curve. And here you got some variation in the detected magnesium. So these are 3 or 4 red experimental traces, which are averaged as the yellow curve with the error intervals. But you see, again, you get a difference between added and detected magnesium carbonate. And it also forms, that is a message, magnesium carbonate also forms prenucleation clusters. We detected them by analytical ultracentrifugation separately. So now you can make equilibrium constants. So you can calculate an equilibrium constants for calcium, and you can do the same for magnesium because you know the calcium concentration in solution, you know the magnesium concentration in solution, and you know how much carbonate is consumed. So all of that is detected. And this is now plotted versus the fraction of the molar fraction of magnesium. So here we got pure magnesium carbonate, here we got pure calcium carbonate. And you see here as the orange curves, it's the equilibrium constant for calcium carbonate formation. And here as the yellow one, we get magnesium carbonate formation. So over the entire range of compositions, you can say that calcium carbonate formation is favored over magnesium carbonate concentration. So it's got a higher equilibrium constant. But nevertheless, the factor is not that high. So that means it can't explain that a 5-fold magnesium excess over calcium excess leads to calcium carbonate formation. So the message here is that although the equilibrium constant is always higher for calcium carbonate formation, nevertheless, the factor is only 1.5 to 4, and it cannot explain the magnesium enrichment in the calcium enrichment in biominerals, because you start from a situation where you have 5-fold excess of magnesium over calcium. So with this, I would like to come to a conclusion. And the first conclusion is that the formation of prenucleation clusters is a new species in crystallization. Their concentration is very low. I would like to remind you, 1% only, but these are equilibrium species. They aggregate and form amorphous calcium carbonate, and if they precipitate out of the solution, they will be reformed. So it's only 1%, but this 1% then can, like in a polymerization reaction, lead to the formation of a large amount of amorphous calcium carbonate. We have tried to characterize the prenucleation clusters in detail, so we worked out their size, we worked out their density. The composition is still unknown. Mass spectrometry is on the way, and Wolfgang Tremel in Mainz reported some mass spectrometry, finding some oligomers at least. We don't know the thermodynamics and the driving forces yet. I've shown you the ITC curve, the calorimetry. What we can say for sure is that the formation of ion pairs and clusters is endothermic as a sum. So that hints towards an entropic driving force. And this is likely driven by the release of hydration water. So I've shown you some qualitative assessment from via density calculations that we have the release of about 18 water molecules by coming from the ions to the clusters. And calcium carbonate clusters aggregate to amorphous calcium carbonate, and this aggregation can be prevented if we stabilize it by silica, and then hopefully we can characterize the structure of the clusters further. Also, the formation of these prenucleation clusters seems to be much, much more common. So I've shown you examples of about 10 different salts, sparingly soluble salts, also as well-soluble salts. We always found these clusters by analytical ultracentrifugation. We are looking for a method to screen for them much faster, to find out whether this is a general phenomenon or not. Also, I've shown you that interactions with additives can be classified already from the very early stages of crystallization. So even prior to nucleation, I can see whether an additive complex is calcium ions. I can detect how many are complex. I can see if the cluster equilibrium is changed, how much it is changed. I can see how much the nucleation point is delayed, what the supersaturation is, and what is the species after nucleation, whether it's more soluble like liquid precursors, or whether it's amorphous or crystalline. And every additive has a very characteristic profile. So hopefully in the future we will be able to categorize that by means of characteristic parameters and then make predictions. So the idea would be we make a titration, and then from the titration curve would be able to tell this additive is going to do this, this, and that. So that would be ideal, but it's of course a lot of work. As you see, I've presented you maybe 5 curves. They are all different, so now you have to do a lot of work and see if you find families of curves and what they do finally. Also, the final example with the magnesium experiment was that if you have mixed clusters of calcium carbonate and magnesium carbonate, the calcium enrichment in the calcium carbonate clusters, which you found as perhaps precursors for biominerals, cannot explain the dominance of calcium over magnesium in biominerals from seawater. So it's not like you have expelling of magnesium from calcium carbonate clusters completely, and these clusters then form the biomineral. So that's not the message, unfortunately. And in the end, hopefully, we will be able to make predictions for titration experiments. So with this, I would like to thank a few people. So first of all, Marcus Antonietti is the director of the Max Planck Institute of Colloids and Interfaces. So he supported me for 16 years, not only with money and useful discussions, but also helped me. Really, he's a great guy. Then also I have to thank Matthias Kellermeier, who did the stabilization experiments of the clusters. Ishi Talmon did the cryo-TEM. Nico Sammerdijk did the TEM on the calcium carbonate clusters. Max Planck Society financed that work. And here are the guys who did the work I presented to you today. So this is Dennis Gebauer, who did the initial work on the clusters. He's now at the University of Constance as a junior research group leader. Andreas Ferch did research on additive-controlled crystallization. Antje Völkel was running the ultracentrifuge experiment. And with a picture of the University of Constance, this is a new place where I am, also close to the lake. I would like to thank you for your attention and happy to take any questions. Thanks a lot.
Thank you very much, Helmut, for a spectacular talk. Jim.
We know that if we make supersaturated solutions that are below the solubility limit of the amorphous phases, we can crystallize. And yet all the titration curves go way above the solubility limit of the amorphous phase. And that says to me that the answer must depend on how fast you do the experiment. So what happens if you do your titration experiments 100 times more slowly?
I think they are already at a lower limit, so if you do them more slowly, they won't change much, but we did them more fastly. And then you're right that you run into the kinetic limit and they supersaturate even more and then precipitate out. So it's certainly speed dependent.
Well, so you just stated that you actually don't get nucleation unless you cross the solubility limit of the amorphous phase.
Right.
But that's not true.
But look, if you look at the at least thermodynamic definition, if you look at delta G equals minus RT ln supersaturation, so if the supersaturation is lower than 1, means you are undersaturated, then you have a positive delta G, at least from the thermodynamic viewpoint.
Well, I guess I was talking about the crystalline phase. You can nucleate the crystalline phase. I mean, we do this all the time, right?
That's true. The crystal is less soluble than ACC, and you can nucleate, of course, a crystal below the solubility limit of ACC.
But your titration curves always go above it, so I'm wondering why you don't ever see nucleation below that.
Why you don't see nucleation?
Of the crystalline phase, right, yeah.
Right. No, so far we also always detected the amorphous phase to be nucleated.
So is there any, so in that case, I was going to ask you a question for which the answer is no. Is there any evidence that you pass through the amorphous phase to get to the crystalline one below the solubility of the amorphous phase? But your answer is you never nucleate until you exceed the solubility limit. Okay, thanks.
So there's something about the ITC experiments that confuse me or that I don't understand. On the one, so you're adding presumably a source of calcium like calcium chloride to a carbonate source.
Right.
That's the titration. That's the ITC, right?
Yep.
So you said earlier that only a small fraction of the calcium is going to be tied up in the clusters, and a large fraction, more than 90%, is going to be tied up as ion pairs. Therefore, the enthalpies you're measuring must be swamped by the enthalpy of ion pair formation, and the whole question of the whole system showing an endothermic heat effect, at least unless I'm thinking wrong about something, would be swamped by what's happening with the ion pairs and their structuring of water and whatever else, and the clusters themselves would be a minuscule contribution, or where is my reasoning wrong?
You're right that most of the system is ion pairs, so only 1% is the clusters. So that's why we try to fit it with ion pairing. So that was the most obvious thing, saying, okay, we have most of it's ion pairs what is forming, so that should be the energy or most of the energy contribution. But in the end, as you can see, they just fit the later stages rather well, but it does not fit the slope, unfortunately.
The difference between the ion pairing and the experiment might give the difference in those curves between the best ion pairing fit, particularly perhaps for the later stages, and what you actually see, that might be what's left over for the cluster formation, and that difference maybe should be what you ought to be concentrating on and fitting,
Right.
that there's real information there.
Yeah. And so far, we were not successful yet. So the models are quite complex. I was amazed how complex the equations are, which you have to fit in the end. But you are absolutely right. Yeah.
Can I try that? Does it work? Yeah, it works. Helmut, I'm curious about the passivation by the silicate of the prenuclear clusters. And I guess there's two possibilities. One is was that you just sort of surround it and have a diffusional barrier, and that would stop it. But since you have so much water in the cluster, I guess it's much more likely, and I'm assuming it's monomeric silicate, that it goes right into the cluster. So you've got a mixed silicate prenuclear calcium carbonate cluster. Have you done any spectroscopy or NMR to try to
No.
determine that?
No, we haven't looked at the structure yet, so it's just known that you can keep them stable. It's on a way to look at their structure, to do spectroscopy, get out their composition and everything. So far, we just, the experiments indicate that the silica must at least be outside, or some of the silica, because if you raise the pH to 11.4, which is only slightly higher than 11, you get them stable and you can isolate them, whereas at pH 11, they are still aggregating.
I see,
So
okay.
that means, because silica is known to be negatively charged, the colloidal stabilization is likely coming from the silica, but it doesn't say that it's not inside. You're absolutely right. So we might change the complete structure of the clusters by combining them with silica. So, for example, the example I've shown you for the magnesium carbonate, calcium carbonate, they also form mixed clusters, so it's not unlikely that they also mix up with silica.
Yeah, yeah.
And this is a problem then again if we seek to determine the structure of the
One
clusters.
follow-up then is how long are the clusters stable once you do that silication? Are we talking about just long enough to sort of make a measurement, or are they stable much longer?
Oh, you can isolate them with a centrifuge and then keep them for weeks.
Okay,
Then.
fantastic. Thank you.
Yes, in nature, we see a lot of different kinds of nucleation going on because of surface catalyzed heterogeneous nucleation. How would that come into play with your experiments? Have you been able to try to take a look at that, or see how that might fit into your scheme of how things work?
We're not able to detect cluster association to a heterogeneous surface yet. We have some experiments, AFM experiments on a glutamic acid crystal which is growing, which point towards that direction. In that case, we had an AFM experiment in solution, and that solution was super saturated, and we saw the surfaces growing stepwise, as you would expect. And then eventually, every now and then, something blocked out from solution, which then spread out, so it really seemed to be liquid, and then ended up in a growth layer, in a single molecular growth layer. Before, it had a height of 5 to 7 nanometers, and then it ended up as about 1 nanometer growth layer. So we think that that could be clusters which attach from the solution. We saw clusters in solution by the centrifuge. We did some recent mass spec, and it showed that we at least got 6 mers in the solution. Maybe there are higher oligomers too. So that seems to indicate that it's possible that clusters fall from the solution, attach to a surface, and then spread out as layer. But that's the only experiment which points toward that direction, which I know of so far.
So if you are in your titration, you go above the solubility of the amorphous calcium carbonate, are the clusters that are forming, are they, if you stop the titration, are you reaching sort of a dynamic metastable point, or do these clusters fall apart as well as form?
We think they are dynamic. And the reason is because we try to separate them. And whenever you try to separate them, they will then precipitate, or if you dilute them, they will fall apart again. So it's a dynamic structure. We try to do NMR experiments to capture the dynamics, but the concentration is so low that the NMR doesn't detect them. So that's, at the moment, it's a problem. It's just speculation that they are dynamic. From the experimental evidence, it seems to be, but we haven't proven it.
So I'm glad you brought up this example of clusters that land on a crystal surface and spread to form layers. So that's observed in a few protein systems if you go to sufficiently high concentration. And those systems have this liquid-liquid separation, and so they're protein-rich droplets. In the inorganic systems, the only example that we've seen is calcium phosphate at very high concentration, where you can start to see these clusters land. But at lower supersaturations, almost all the growth is ion by ion. So I'm wondering if that's the explanation for your magnesium results. Is it possible that the clusters are only important in this initial nucleation event, but once you pass a critical size with all those ions around, the thing is going to start growing by adding on ions. Right. And in that case, the kinetics of calcium versus magnesium attachment at steps becomes the dominant factor in determining the concentration. Just wondering what you think, sort of the relative importance of clusters and ions.
Well, what you see from the titration curves is clearly that at the point of nucleation, the ion concentration clearly drops, and after that, that means ions are consumed. So the nucleation is not only driven by the clusters, obviously, otherwise you wouldn't detect the ion drop, obviously. And if you look at the curves, calcium versus magnesium, they behave differently. So it's also different importance of calcium versus magnesium to get into the mixed clusters. But once they nucleate, you have the amorphous phase, then you get this constant region of the curve after the nucleation point. That means ions must attach to the surface because the concentration of ions stays constant. As you know it from the textbook, it's a solubility product, and it stays constant all the time. That means everything you add must be incorporated into the solid precipitate, whether it's crystalline or amorphous. I absolutely agree.
Okay, I have a comment about magnesium too. Lori Gower and I did some measurements of growth rate of ACC films with the polyacrylic acid and with and without magnesium. So another ingredient is the hydration shell of the magnesium is probably harder to disrupt than of calcium, and that would have to be an ingredient in how fast things happen. Yeah. We found that magnesium caused these kind of delays of incubation time. So I would be really interested in your work if there's a correlation between presence of magnesium or anything. I'd be interested in anything that causes these delays before the titration curve starts to rise. That may tell that there's a delay in getting one ion out of being associated either with water or with the polymer additive.
You definitely detect them quantitatively. I haven't shown the overlay, but if you look at curves of the delay of nucleation versus magnesium concentration, you can scale that. And that means you definitely got a dependence.
Then you saw the same thing we did, that's right.
Mm-hmm.
Okay.
Yeah, I think that previous question posed the same question that I was going to. My understanding is what happens is that in seawater, you are likely to get the formation of clusters which contain both calcium and magnesium. And presumably because it's very much more difficult for the magnesium ion to desolvate, these clusters are going to essentially desolvate the calcium ions, not the magnesium ones, and so progressively you're going to lead to the formation of, say, precursor vaterite or maybe calcium. So doesn't this actually constitute the basic explanation as to why you see no more than, say, 18–20% Mg substitution in biogenic calcites, and that you're not likely to ever to see magnesium as a predominant, magnesium carbonate as a predominant component, because you're never going to get beyond nescaronite or Lansfordite or these hydrated magnesium carbonates.
Well, that's certainly the point, that magnesium is much harder to dehydrate. And that was also something we had in mind when we did this experiment or designed these experiments. We thought maybe magnesium is so difficult to dehydrate that calcium is so much more favored over the magnesium. But in the end, if you look at the curves, the difference is not that high. So it was up to a factor of 4 only, if I remember. So here we are. So factor 1.5 here to 4. So certainly you favor calcium all the time over the entire magnesium concentration over magnesium carbonate, and the explanation will certainly be because it's easier to dehydrate calcium rather than magnesium. But at least going through the clusters, it wouldn't explain the dominance, the effect that calcium is so much more favored. Because otherwise you would expect that the clusters contain much, much more calcium than magnesium than what we find. So there's still surprisingly high content of magnesium present here in the system.
So just to return a point that was raised by Jim, I'm wondering, you've, I think, implied in your discussion of nucleation that essentially that happens through aggregation of these pre-nucleation clusters. But I'm just wondering if they could really be acting as seeds, and that the nucleation event really occurs through ion addition onto those.
Very likely indeed, because you got that drop, and told that to Jim also, you drop at the nucleation point. So what you have at this point is that you have the aggregation of clusters which already occurs before the nucleation point. You get the macroscopic precipitation at that point, if you look at it with a microscope, but also the drop in calcium concentration. So it's very likely that the ions must add to the precipitated species because solubility remains constant. So it's, if you wish so, it's a seed, yeah, which grows by further ion addition.
Yeah. So I have a question about the fact that you put the adjective stable before the prenucleation cluster. You shown these two graphs at some point in which you show a metastable cluster and then a minimum in the energy. I wonder what is the piece of information that tells you that this is a stable cluster and it's not just a result of a stochastic process?
The thing is, Dennis was doing equilibrium thermodynamic calculations on the clusters, and you can describe them by equilibrium thermodynamics. So you applied mechanisms known from protein aggregation to describe protein aggregates, essentially, and they also describe cluster formations. So, but to point out, they are not stable towards the amorphous calcium carbonate, which is formed later, because otherwise it wouldn't form, but they are stable towards the ions, which are there initially. So this must be distinguished. So thermodynamically stable just means I have a minimum, and this minimum is lower than the initial stage. But I got another minimum, then it's towards unstable towards the next one. So we have to distinguish between these two situations.
I'm not an expert in this field. I just have a quick question about the buffer you use to keep your pH. So you use sodium hydroxide.
Yes.
Would you expect any changes if you used potassium hydroxide instead?
Oh.
Because we see in simulations that sodium ions associate pretty strongly with carbonates
Okay.
in solution, but potassium does not.
Right.
And I wonder if it would have any impact on the initial growth of these oligomers in solution if you have to compete with another ion that has to be displaced to form this calcium carbonate
Yeah.
cluster.
I can't answer you this question because we simply didn't look at it.
Uh-huh.
We got a lot of sodium in there because the carbonate is sodium carbonate we start with, or sodium bicarbonate.
Okay.
So we certainly have the counter ions, and we use sodium hydroxide. So I don't know what happens if you would do the same with potassium carbonate and adding potassium hydroxide. You would think that that could make a difference? So it
We
would
don't
be
know.
rough?
It's just that we never see strong association between potassium ions in solution and carbonate in a pure potassium carbonate solution, but we do for sodium.
Okay.
It's just
So
a
maybe
thought.
worth to perform such experiment. Just never thought about it.
Okay.
Okay, I have a question of my own. So carbonates are somewhat special in that they are more soluble at lower temperature. So I'm wondering if you started working on calcium carbonates, and I saw in one of the slides, if you can actually go back where you showed there was cesium iodide and a few other compounds. So clearly the precipitation of calcium carbonates is entropically driven rather than enthalpically driven. I wonder if the other compounds for which you observe prenucleation clusters also fall into that category or not.
Likely not. If you look at sodium chloride, for example, that's a well-soluble system, and I think it dissolves with higher temperature, so it's opposite of calcium carbonate, and it also forms the clusters. I think the same is true about ammonium acetate. So it seems to be more general. It's not peculiar about carbonates, which surprises us a lot. So we expected something different. So we thought it's not forming for sodium chloride, but it did.
One last brief question. Can you give me a quick idea of the concentrations at which you start seeing prenucleation clusters, and how do they compare to saturation?
Yeah, that's a good question because so far we detected them mainly by analytical ultracentrifugation, which works with refractive index detection. The calcium carbonate, we work with 10 millimolar solutions, so it's about that range. Yes, and you see, for example, it is an issue to detect them. So if I look at the centrifugation data again, so you see that in the initial stages, if you look at the red curve, I don't see any clusters, although I've shown you that the clusters are there straight away in the titration curve, so you see the difference straight away. And also, if Nico looked at it only 1 minute after you put the solutions together, oops, so went over, but he can see the clusters already. So at the very early stages, so they are there, but you can't detect them in the centrifuge. So this is certainly the undersaturated case still. So
Can you translate that into units I understand?
It means 1 minute is, if you constantly add, then let's go back to titration curve. So 1 minute is 60 seconds, somewhere here, at the very early stages.
So that would be what, a tenth of saturation?
Yeah, completely undersaturated, I would say. Relative to the concentration of the final species, which is precipitated. So that defines our solubility as amorphous calcium carbonate, which is precipitated. Mm-hmm. Yeah.
So it's 10 times the solubility of the crystal.
Yeah, way off. So it's somewhere here. If you look at that, this is 1,800 seconds, so 60 seconds will be somewhere here. It's not even resolved.
So if all of this is true, and I'm perfectly happy to believe your excellent data, this is a new kind of chemistry. So the usual, we have a bunch of ions in solution, and from that everything starts, including life, including any precipitation of any crystal on Earth,
Right.
should be rethought in terms of prenucleation clusters.
Well, yeah, but the concentration is low, that's one thing. But they're equilibrium species, so as soon as you take them out of equilibrium, they will be reformed.
That's another thing I haven't understood. The concentration is low with respect to what? You said 1% of what
Of
exactly?
the ions, the total amount of ions. So what I detect in the centrifuge is refractive index. So this is the sum of ions plus ion pairs plus clusters. And if I separate the clusters out, then I can only detect a refractive index. And if I assume the refractive index of the clusters being the same as the ions, otherwise I can't relate the concentrations, then I end up with this 1%. So this is the minor amount.
So that confuses me about the statement about stability, coming back to Alex's question. If the number of clusters are only 1% of the number of ions, then the free energy of the clusters is higher. It's higher by log of 100. It's still lower than all
And
the
the
other
ions.
sizes and the
Yeah.
bulk, but it still
Yeah.
must be above the free energy ion pairs, and that's why they represent a small fraction of the total
And
number.
this you deduce from the small equilibrium constant by saying I got more ion pairs than clusters, so that means the constant is smaller than 1, and that means the energy must be higher. Yeah.
So they're stable in this, in the thermodynamic sense that there's always a population there.
Yeah.
But any one of them is higher energy than the ions.
Right.
Well, yes.
I imagine the interfacial free energy of these clusters must be exceedingly low, isn't it?
They
Do
even
you
don't
have
have
any
one, I
idea
think.
what magnitude is of the interfacial energy?
If you look at the curves, the clusters are no particles, they are solutes. So you can more compare them maybe like a dissolved polymer rather than a particle. And I deduce this from the fact that the calcium concentration or activity is still increasing if I add calcium, whereas here in the two-phase system, after nucleation, where I have particles, it remains constant. That means I must have, the clusters are soluble before, so they shouldn't have any interfacial energy.
Well, thank you very much again.
Thank you.
Cafeteria, and anybody who's interested is welcome to join for further informal conversations. Thank you for attending.
They were happy with pretty much anything you tried forms prenucleation clusters.
That's true.
I have a very, very fundamental and deep disagreement with you that we have to work through.
That requires a glass of wine, doesn't it?
Yeah, well, we're going to both be in New York next week.
Plenty of wine.
So I think it's just simply not true that these crystals won't nucleate below the solubility limit of the amorphous phase. You can set up a solution that's undersaturated.
Sorry, I didn't say this.
But
So if you have
the titration data will show it.
but if you perhaps would titrate them so slow.
That's what I'm asking.
Yeah.
So if I go 100 or 1,000 times more slowly, these curves must not go up as high because I know I can precipitate below the solubility of the amorphous phase. Okay, so that's point number 1. The second question is it really a pathway through the amorphous phase? If I nucleate the crystal directly, without crossing the solubility of the amorphous phase, granted, I firmly believe that the clusters are involved because I can't explain our data without invoking some kind of magic or without invoking some kind of way to avoid adding ions on to make a big piece. But the very, very first nuclei, and we're talking about things that are sub 2 nanometers already are crystals. Whereas, and you can say, well, that's a problem with the experiment, except we know that if you cross the solubility with the amorphous phase, the very first nuclei are amorphous. We using exactly the same technology. So there's something missing here that we've got to sort through. Because this whole picture, which is, you guys have proven this, certainly to my satisfaction, but it's all in a regime that's way